Good morning, ladies and gentlemen. It's a great pleasure for us, for Dr. Hans Kreis and myself, to chair this session. And we would like to welcome all of you, and we are happy to see so many people coming on the last day, and especially the day after the night before. So thank you very much for attending this early morning meeting. We shall start with the David Neve lecture. For those who don't know the story of our federation, I just say that Professor David Neve was one of the pioneer of founders of the European Pain Federation. Dave Neve devoted his life to the development and the promotion of pain medicine and even the concept of pain medicine. Dave was a director of the Center of Pain Medicine at the Zuratsky Medical Center affiliated to the Tel Aviv University, and by this way he became a founding member and president of the Israel Pain Association. But he was also very much involved in the European development of pain medicine. He was the president of EFIC and was founder of the Europe Against Pain Initiative, and also on the development of the first European Week Against Pain in 2001, which then became the Year Against Pain. And finally, together with Marshall DeVore, they developed the EFIC declaration that chronic pain is a disease on its own right, which is one of the cornerstones of what we know today as the found of pain medicine. So for me also it's a special honor and pleasure to introduce you today our speaker of this David Neve lecture, Dr. Vania Apkarian from the United States of America. Because with Vania we have got a long-term, deep, and rich friendship. At the very beginning we shared our common passion of the study of the functional architecture of central pain pathways in animals. And at that time, we were considered as a sort of old-fashioned people that tried to follow the path from Don Santiago, Ramon y Cajal. And we support each other, and I always got the Vania enthusiasm to continue. And today you will see that which shows clearly the strong impact of these seminal studies on the work that Vania is doing today. Vania Apkarian is a professor in the Department of Physiology at Northwestern University Feinberg School of Medicine, and his work focuses on pain perception in health and disease as a means of studying consciousness. He has pioneered the use of brain imaging technology to peer into the brains of individuals who suffer from chronic pain conditions, resulting, as you will see today, in seminal observation how brain function and structure adapt to the chronic presence of pain. Vania's more recent work has included the first prospective prediction of pain chronicity using brain properties and the first description of brain activity that predicts placebo responses in pain patients. Uh, Vania Apkarian's current focus is the use of brain imaging in animals and humans to determine the critical mechanisms that drive the propensity for developing chronic pain, its long-term maintenance, and therapeutic interventions aiming at treating and ultimately preventing the development of chronic pain. So please, mon ami, I'm so pleased to up your hands.
Thank you very much. Thanks. Well, thank you for having me. This is a wonderful venue, and I'm delighted to address Ethics Society. This is the first time I'm attending it. Hopefully, it will not be my last. Thank you, Luis, for those wonderful introduction. Luis and I go back a long time, and yes, it's a very old friendship. And I should add that besides Ramon y Cajal, I also would like to include in that list Golgi as well, just to, add the both extremes of thought of how the brain neuroscience works in general. I want to address, to give you an overview of, in a sense, where we are in our concepts of what are the mechanisms underlying chronic pain. This is a quest, I suppose, that we have been pursuing for more than 15 years, and it is humbling, in a sense, how complicated the mechanisms are, and there's a long way forward. But there's a very simple concept behind it, which I think we have proven it to be valid, and I will demonstrate it to you in the rest of my talk. And the idea is very simple. To understand mechanisms of chronic pain, we have to understand the clinical condition itself. We have to study the patient. And whether you like it or not, currently the only mechanistic approach to study the patient is to look inside the brain of these patients. So currently, this is the approach, and the approach, in a sense, is almost a reverse translational approach, so that we look inside the brain of the patient, then we look at its correlative mechanistic models in animals, and hopefully that would then translate into a forward translational ideas that then we bring it back to the clinic and test novel therapies. So that's the general idea, and let me give you a little details on the subject. So, again, old people with lots of white hair put up old slides, and this is my favorite slide. This is the Descartes' view of what is sensation, in fact, in general. And obviously, this is very dear to pain scientists in that it captures the idea of perception in general from the viewpoint of pain. Descartes, back in 1644, illustrates transduction, transmission, and perception all in one diagram, which is fantastic. And in a sense, over the last 100 years, all the research that's been done in our field has concentrated at identifying the components of this system. There are many important unanswered questions. I have sort of listed them here. I will sort of indirectly touch on them, given the amount of time I have. But overall, at least for us, there is a new challenge of dissociating sensation and behavior from each other. This is an idea that I will come back to at the end of my talk. There's a big debate in our field about localization versus integration. This is the debate between Ramon y Cajal and Golgi, and we are still battling it, at least in our field, right? And again, the other issue is, what is the scale? At what level do we understand perception, and at what level do we understand pain? Pain as being a conscious state of beingness puts very hard conditions about how to really integrate its understanding. And then the main thrust of my talk today is the relative role of the end organ injury site versus the brain-gene interaction regarding risk of developing chronic pain. So within this general overview, let me move on and simply recapitulate things that you know even better than I do. The definition of chronic pain is tautological. We say it's chronic pain if the pain persists. That doesn't say anything except that there is pain, right? And perhaps it is agnostic to its mechanisms, and in that sense it gives us an opportunity to explore mechanistic overviews. But the field, due to a lack of any other opportunities, has been dominated by sensation itself. So the basic research about pain has heavily concentrated on these ideas of allodynia, hyperalgesia, central sensitization, and its clinical parallel has always been this, again, whether the injury properties are neuropathic or inflammatory, the extent of central sensitization. All of these are concepts about the sensory processes underlying the pain, and it is not obvious that it actually gives us anything mechanistically profound one way or another. In fact, I would challenge that idea. Now, again, coming back to this sensation idea of pain, now we all know that as soon as an injury persists and there is some local persistent inflammatory mechanisms, there's a massive reorganization of the afferent fiber system with a whole variety of its specific receptor types that are upregulated and or downregulated. Here's the spinal sensitization cartoon that I'm sort of just illustrating. So we know that these things are reorganizing. And over the last 15 years or so, this is what we have been sort of advocating, in a sense that the cortical mantle itself is undergoing both anatomical and functional reorganization due to chronic pain. Now the literature along these lines has become in fact quite vast. I will not review all of it. I will concentrate mostly on our more recent work on the topic. But now we have hundreds of studies showing that brain anatomy and brain function are now different and specific for different types of chronic pain. So for the rest of my talk, I want to put up a new definition of what is chronic pain and try to defend it. This is a viewpoint obviously biased from our own work, but it is it's the viewpoint of a behavioral selection, that how one makes choices in the environment with and without the presence of pain. And the fundamental idea is that pain that engages the emotional learning circuitry and reorganizes the value of pain itself is, in a sense, a mechanistic viewpoint of what is a chronic pain state. Emotions and brain learning circuitry and reorganization are the three fundamental elements that I'm introducing into the definition of the functional definition of chronic pain. Here's my outline of approximately what I want to talk to you about today. The important points that we have made, and now many other people have made as well, is that acute pain and chronic pain are distinct chronic pain states. Brain states, not pain states, sorry. And different types of chronic pain are specifically, they have their own signature, both anatomically and functionally. I will show you some of our longitudinal data where we actually track subjects as they transition from acute to chronic pain, and in that set of data, I want to emphasize that the limbic brain properties are the primary risk factors for chronic pain, and it underlies a cortical, subcortical, limbic, cortical learning processes. Finally, I'll show you some data about more recent rodent studies, animal studies that are complementary to these essential concepts, and in a sense that we are seeing a cohesion of ideas now from the human brain imaging studies going back to rodents, hopefully moving forward from there. So this is my single slide claiming that acute pain and chronic pain are distinct states. The panel here on this side is a meta-analysis of 224 brain imaging studies. Fundamentally, these are acute pain brain imaging studies, and the picture that you see here are the brain areas that one identifies associated with acute pain. This is a meta-analysis. As a result, you can think of this as the average of about 2,000 subjects having either an acute thermal or a mechanical stimulus. The panel on this other side is brain activity in some 15 chronic back pain patients. And the point I'm trying to make is that the region that we identify activated, this medial prefrontal cortex, is not even here. So either that we are doing something very stupid that has nothing to do with pain and studying some epiphenomena, or that acute and chronic pain are distinct. So I will spare you all the details of all the evidence, but the evidence is quite compelling that these two systems become independent. Again, now more than 10 years ago, we have shown that the brain anatomy itself is distinct and is reorganizing in chronic pain patients. This was the first evidence showing that dorsolateral prefrontal cortex and thalamus gray matter density is decreased in chronic pain patients. Similar studies soon after us was done in fibromyalgia at Medial, in tension headache. This was in Germany, I think. Irritable bowel syndrome, this was in Toronto. These are very early studies. There are now hundreds of such studies showing a very complex pattern of the brain anatomy in chronic pain patients. And suggesting that it's uniquely different for each condition. So let me show you some of the evidence that we have generated specifically asking this question. This is work done together with Marwan Baliki. Marwan and I have been working together for more than 10 years, and almost all of our seminal brain imaging studies were done with his leadership, and he's now starting his own lab. So this is a complicated picture, but let me simplify it and see if you can share the excitement when we generate these pictures. So this is a heat map, basically of all 80 brain areas, and it simply shows the self-similarity of the regional gray matter density. The areas that are red next to each other are more similar in their density, and the areas that are blue are less similar. Okay? And this is across some 25 or 30 healthy subjects, which simply shows the gray matter spatial similarity. Okay? A heat map, a simple heat map. If we generate the same heat map exactly with the same technology in chronic back pain patients, complex regional pain syndrome, osteoarthritis patients, I'm not going to give you any statistics. You can see visually that they're different from each other. Right? So the self-similarity of the brain as an overall, global idea is disrupted in chronic pain patients. It's disrupted in unique ways in different types of chronic pain. Okay? In fact, if we take that as an index, as a global index over the whole brain, as a single number, and plot it with respect to the number of years each subject is suffering from pain, we generate these wonderful exponentially rising curves, basically showing that in chronic back pain patients, the brain is reorganizing over a time scale of about 12 years. In CRPS patients, that reorganization is more rapid. It plateaus by about 1.5 years. In osteoarthritis, it's something in between. We cannot exactly measure it. But So there's a massive reorganization. We don't understand all the details of this reorganization. It is happening, and it's uniquely specific, both in its locations, in its overall impact, and its time constant. So, just to move on, the data that I have showed you so far is, in a sense, is all cross-sectional. It is comparing subjects, different chronic pain conditions, to healthy subjects, and looking at the differences. But the critical question is that clinically, most of you at least know that you can see exactly two patients with the same type of injury. One of them develops chronic pain and the other does not. So what is the difference between the two? And the only way to untangle these things is to do longitudinal prospective studies where we can actually follow the subjects and look at how the brain is evolving in time and whether it has any relationship with the outcome, with the clinical outcome. So we undertook this study now 6 or 7 years ago. It took us about 6 or 7 years to even collect the data. So this is a massive undertaking, and still I think it's the only longitudinal brain imaging study probably in all of neuroscience, and specifically for chronic pain. So this is a longitudinal, cross-sectional, observational study. We recruited subjects with acute back pain, of pain that was of 4 to 12 weeks in duration, with no history of back pain for at least 1 year before that. So in a sense that an acute new back pain event, and back pain of enough intensity that it would give us the possibility of studying its transition to chronic pain, so pain of 5 out of 10. So we would monitor a whole variety of brain parameters, basically anything we could think of we would look at in the brain, and then as the patients transition to either recovery or to chronic pain, we ask the simple question, what is the role of the brain? Right? So here's an overall conceptual design. We call these subjects subacute back pain. They come in at 5 different visits to the lab over the span of up to 3 years from entry into the study. We scan anatomically and functionally their brains, and in time, these subjects either persist with their back pain, so these are becoming chronic pain patients, or they are recovering, so we end up with 2 subgroups, and then look at either events that are changing in time in this time period. Those are the consequences of developing chronic pain, versus events that are constant and exist at time 0. Those are predictors or risk factors for chronic pain. So here is the simple pain properties of this population. We were in many ways lucky in that essentially the population subdivided in time 50-50 into 2 groups, persisting and recovering. So, and in time, so this is their back pain intensity, VAS score of back pain over 1 year. The subjects who are persisting with pain, their pain never changes. Okay? So they started at around 6 out of 10 pain. A year later, they're still at that level. The ones who are recovering, they're recovering relatively fast, but they continue to decrease in their pain. And you can see 1 year later, there's a massive difference between the 2 groups, right? And in fact, of the population that we follow up to 3 years, this difference is essentially the same. So 3 years later, about half of the subjects are still in the same level of pain, while the other half have decreased by at least 50% in their intensity of their pain. Let me skip on this slide just for time. So if we look at the brain activity in these subjects, what do we discover? I have subdivided the brain activity into the recovery group, the SPPR, the recovery subjects, the persisting group, and in each case, I'm showing you the group map for brain activity, either at visit 1, visit 2, visit 3, or visit 4. At visit 1, this is the brain activity for the subjective perception of pain. Okay, and that's an important detail which I will not elaborate. We can see a robust brain activity with matches between the two groups. The two groups are not different from each other in their brain activity for their pain state. And importantly, that map matches very nicely the acute pain map that I showed you about 2,000 subjects. So in a sense, we can say that initially these subjects start with an acute pain state. Okay? And over the 1 year period, the subjects who are recovering, eventually, at 1 year, there is no more brain activity. We cannot detect it anymore. The pain is low enough that the brain signal is simply not robust. We cannot capture it. The patients who are persisting, something very dramatic happens. The brain activity in the sensory areas continue to go away, while this medial prefrontal cortex, bilateral amygdala regions become higher. So there's almost a complete shift away from sensory regions of the brain being activated to emotional areas, which in fact matches nicely to my initial picture that I showed you, which is our first study that we did, in a sense, almost 15 years ago, trying to understand what was the brain regions that are activated. And here we see that longitudinally, in a sense, these subjects are shifting in time away from sensory areas of the brain to emotional areas of the brain. So that's a very curious result. It shows that there is functional reorganization of representation of the pain. And what are the mechanisms behind it? Let me elaborate a little more. Now, in the same study, I'm not going to show you all the details, we identify two areas of the brain. Again, the medial prefrontal cortex and nucleus accumbens. And the amount of information that's shared between these two areas is high at visit 1 in the subjects who are persisting, and it's low in the subjects who are recovering. So this is at visit 1, right? Even though their brain activity for their pain was exactly the same. And if we look at the same parameters at visit 2, visit 3, and visit 4, we see exactly the same thing. So here is a signal that a priori, at time 0, differentiates between the two groups and is constant over the duration of the study, at least over the 1 year. So we think of this as a risk factor, as a predictor of chronic pain. Let me say a few words about this network. The medial frontal cortex accumbens is part of the mesolimbic system. It is part of the network that has to do with addiction, with alcohol addiction, cocaine addiction, with negative mood processes, with learning and motivation and behavioral modification. So this is not just another area in the cortex, this is a fundamental circuitry that has to do with the dopamine signal that is then in turn modulates the brain. I'll come back to that again at the end of my talk. And so this is very provocative, and in a sense it's suggesting that the brain circuitry is reorganizing in some of our subjects into becoming addicted to the pain. Very simple. Okay, and we've pushed this idea for a number of years now, and very recently there are now 3 different animal publications that really nicely complement this data. I'm only showing you one of them. Here's a study by Lee et al. That was published in April this year in Journal Neuroscience. Basically, these are channel rhodopsin optogenetic studies where if you activate channel rhodopsin in the medial prefrontal cortex in rats following a neuropathic injury, you can completely reverse, at least for a short time period, the neuropathic injury allodynia. This is the allodynia in the SNI animals, and if you activate the channel rhodopsin, it completely reverses it. Similarly, it dramatically decreases the cold allodynia. Now, in the same study, when they actually look at the projections of neurons from the prefrontal cortex to accumbens, and now activate with, again, light, the channel rhodopsin terminals within accumbens, they essentially recapture the whole thing. So this system seems to be able to at least modulate only neuropathic animals, but not in healthy animals. There is no, one does not see this. And there are two other studies essentially confirming the results. I highlight this paper because it is, for the first time since 1969, we actually have a new target in the brain where we seem to be able to specifically modulate chronic pain as opposed to acute pain. Since 1969 was the discovery of descending modulation, opiates, induced analgesia. Here is, in a sense, its equivalent for chronic pain. It's very exciting to me because in many ways it really, confirms our human studies. Let me go back to the human studies. Now, in the same study, we have also looked at the brain white matter properties, basically how the myelin properties of the cables connecting different areas of the brain to each other are maybe distorted in our subjects. And we can see these are all the red dots here are areas where myelination seems to be thinner overall in the subjects who are persisting in pain. And this picture, in fact, we identify at time 0 again, at the beginning. And again, if I plot this myelination abnormality, we can see at time 0, and it's persisting across all times. So it seems like the axons, the properties of the axons in the brain, to start with, are already abnormal in the subjects who are developing chronic pain. Now, very briefly, let me show you some of the additional data we have recently generated, and this is part of a study that's submitted for publication. And essentially, looking at the same subjects now, up to 3 years after the initial event. And again, at 3 years, essentially, the pain properties are still distinguished between the two groups. And now if we actually study just the mesolimbic brain as a circuit and look at its properties, we can subdivide it into 3 different parts, one of which, this prefrontal cortex accumbens amygdala network, seems to have both anatomical and functional properties at time 0 that distinguish between the two groups again. So this in some ways confirms the earlier observation, but this is now sustained over 3 years. If we now look at simply the size and the shape of the amygdala and the hippocampus, again, we see that in fact the size of the left or the right hippocampus is smaller in subjects who are developing persistent pain. The ones who are recovering have the same size as normal subjects, and the difference is sustained over 3 years. We see exact same picture for the amygdala also. So amygdala volume is again smaller both in the left and the right in only the patients who are becoming chronic pain, and these differences are sustained over 3 years. And in fact, if you actually look now at the morphology of the amygdala and the hippocampus, we can actually see that across the whole at least 1 year period, there are regional abnormalities in shape that distinguish, again, persistent from recovering. So, to summarize a bit where we are, I have shown you that this network itself, the prefrontal accumbens amygdala network, functional connectivity and white matter connectivity, and its size together, all of them are risk factors for chronic pain. So what happens if we actually build a model like this? Here's my model. The model, in fact, shows that we can explain 90% of the variance for risk of chronic pain by these 3 parameters: brain white matter, functional, and volume properties. Each one of them are independently significant as far as predictions. How am I doing on time? 2 minutes. Okay, all right. Let me just skip that. Now, if instead I build a model from specifically for the symptom severity, how much pain do subjects have 1 year later, I see a very different picture. Remember, the initial contrast was how much change was in their pain. This is how much is their pain, and we see in fact that these brain parameters have very little effect, and it's primarily controlled by the pain at time 0. So here is an overall picture of what I'm trying to convey to you, that we have a predisposition, brain parameters, their interaction with the injury gives rise to this learning mechanism, which in turn gives rise to the chronic pain state. Let me just, for a few minutes, just show you some of the animal data, and then I will finish my talk. We have now been imaging rats, both under anesthesia and awake, and again we see following an injury that there's a massive reorganization of brain circuitry which takes time to come about. The reorganization involves specifically nucleus accumbens circuitry, and now if we take the tissue from those animals, we can see that the accumbens receptor expression is changed, and that change is in fact related both to functional properties and to behavior as well. When G. Ren presented some data to you in a poster a few days ago, of our physiology of nucleus accumbens, where we actually show neuronal response properties changing in time in a very elegant way, and this paper should be coming out soon as well. And again, recapitulate the data that I just showed you earlier about channel rhodopsin studies. And importantly, we have now been showing that the hippocampal neurogenesis is an important and maybe critical factor for the transition to chronic pain, saying that hippocampal learning processes are also part of the circuitry that's involved in chronic pain. So here's my overall summary, that the mesolimbic system involved in reward, addiction, negative mood, and now in chronic pain is part of the circuitry that we have to incorporate in our concept of what is chronic pain. This circuitry is receiving and controlling nociception as well as the cortex. If we have an injury, then the injury in turn may in fact give rise to this cortical control of emotional learning, which would basically help subjects recover from that initial injury. On the other hand, if the mesolimbic system is predisposed to a larger emotional learning signal, then it will, in fact, through hippocampus, amygdala, accumbens circuitry, then in fact change the cortical representation of that pain and create this chronic pain brain state. And the equivalences between the human and the rodent are in fact striking, and it gives us a wonderful tool with which we can go back and forth between them to really begin to untangle the specific mechanistic processes. To summarize, since brain characteristics are determining propensity for chronic pain we have to consider it as a neurological vulnerability. Okay? And the limbic circuitry are the primary risk factors for chronic pain. And the limbic nociceptive interaction is the signal with which the cortex is being reorganized into unique, specific chronic pain conditions. So, and overall, I hope that you appreciate that these concepts should give us new tools with which we can begin to develop both preventional and treatment strategies for chronic pain. Let me just finish by saying that the future of this field has to be with multi-center collaborative approaches where we bring the clinical patient into the basic science and we incorporate the brain science with the clinic, and to that end we are trying to create this open data sharing system that I would like everybody to participate in, and I hope you drop us a line and say you are willing to be part of this, because we need the help both of the clinic and all the scientists involved in brain imaging. And lastly, I'm the speaker. Many people have put huge numbers of hours to make these studies to happen, and I should thank them and NIH for supporting these studies. Thank you.